Hi, I am Dinesh Kumar. So I'm going to present to you my poster titled A Protocol for Studying Metabolic Interactions in Microbial Communities Using Graph-Based Approaches with a few slides that I have prepared. The main motivation behind undertaking this project is studying microbiomes. Microbiomes are complex systems. In order to understand them, we need to study how the microbes interact with each other in a microbiome. But replicating these microbial systems in laboratory conditions is not very easy, so there is a need for computational methods. These computational methods should find interesting features about a community, it's thereby helping to focus wet lab resources to get a very desirable outcome in a short period of time. So to attain these goals, we need a method, computational method that uses only the available genome information and is also scalable to accommodate large community networks. To achieve this task, we use something called as genome scale metabolic networks. These are bipartite networks. They map the whole topology of metabolic reactions in an organism. These can be constructed for any organism given we have the genome information. In this protocol, we use this special Python package called MedQuest to work with these metabolic networks. Before going to the method, let us see what to expect from this protocol. This protocol will help us predict possible interaction patterns in the microbiome. It will help us capture changes in interactions with change in environment. It will help us see the possible metabolic exchanges that can happen in the microbiome. It will help us see the dependence of one microbe on the community, and we'll be able to predict possible metabolic capabilities of microbes in the given environment, and how it affects the community, and we'll also be able to come up with some hypothesis on the working of this microbial community. This is the overview of the process. First, we start with genome scale metabolic models. This is done by using Python package called MedQuest. MedQuest builds genome scale metabolic models from genomes of microbes present in the microbiome. Networks are built for individual microbes, for microbial pairs, and for higher order communities also. Further, MedQuest allows us to find active and inactive reactions in the metabolic networks for a given media conditions. These media conditions are represented by a set of metabolites chosen based on the environment in which they are obtained from. We see that the number of active reactions increases when microbes are present in community versus when present individually. This represents dependencies of one microbe on the other. We use two scores called metabolic support index and community support index for predicting this, quantifying this metabolic dependency in microbial pairs as well as in higher order communities. By looking at overlapping exchange reactions in the metabolic networks, we'll also be able to predict possible metabolic exchanges in the microbiome. Further, for higher order communities, we will also be able to predict something called as unique contributors. These are microbes that have very unique metabolic capabilities in the microbiome. With this protocol, we studied two different microbiomes, hydrothermal microbiome and ocular microbiome. First, let us look into the results of hydrothermal microbiome. Hydrothermal vent microbiome data consists of 98 metagenomically assembled genomes representing corresponding to 98 different microbes. So this has a combination of both archaea and bacteria. So we were interested in looking at the interaction between archaea and bacteria. So we constructed different different metabolic networks for this microbiome. This is one of the results that we saw in hydrothermal vent microbiome. So Paciorchata is an archaea present in this hydrothermal vent. So this figure represents the interaction pattern of this Paciorchata with other microbes in the community. So you can see that all the arrows are going towards Paciorchata, which means that this Paciorchata is capable of obtaining support from all the other microbes in the community, while it can only provide very little support to other microbes in the community. This could mean that Paciorchata is one of the most dependent microbes in the microbiome. We also studied ocular microbiome using this protocol. This data had two different communities in it, a healthy community and bacterial characteristic infected community. So we wanted to see what are the differences in microbial interactions between these two communities. The networks present here represent the interaction pattern of healthy community in two different conditions. The first one is in the healthy condition, the second one is in the infected condition. These edges represent the interaction between two microbes. The thickness of these edges are mapped to MSI scores. The higher the thickness, the higher the MSI score. You can see that in healthy conditions there are a lot of interactions, while in the infected conditions there are very less interactions. This might mean that in infected condition, the healthy microbiome has very less potential to interact with one another. Finally, we can conclude that this method can help predict interesting features in a community, like key microbes in a community, effect of change in environment on the microbial interactions, the metabolic capabilities of the community, and etc. So, of course, there are some limitations in this approach. This metabolic networks are only as good as the genome that we use. So it is important to have a good quality genome, and there is a requirement for the suitable representation of the metabolic environment to have accurate results. Finally, I acknowledge Dr. Karthik Raman, my advisor for this project, and Dr. Karthik Anandaraman for providing the hydrothermal data, and Dr. Shivaji for providing the ocular data, and my lab mates, and Ministry of Education India for supporting my master's studies, and ISMB travel grant for helping me attend this conference. Thank you.